Okay, so temperature is the big topic we've been doing, and I have with me here Professor Miles Lamar. And Miles, would you like to introduce yourself and tell us what you do?
Yeah, g'day, kia ora everybody. I'm Miles Lamar. I'm at the University of Otago in the Marine Science Department. I've got a research interest in the effects of climate change on marine species, and in particular I'm interested in knowing how Antarctic species are going to respond to warming. Antarctic is a super interesting place from a biological point of view, but it's also a place which is going to undergo quite a bit of warming over the next hundreds of years, and so we really are interested in knowing how polar species are going to respond to warming. What kind of capacity have they got to adapt? What might the ecosystem look like in another 100 years under a place which is maybe 3 or 4 degrees warmer than it is today?
So, Miles, we're basing these predictions on the increase in temperature. In fact, a destabilised equilibrium with more energy being retained in the Earth's surface and atmosphere than we're losing. And this is being taken up by the ocean, is that right?
That's right, yeah. So the ocean has got a huge capacity to take up heat. One of the differences between land and sea, of course, is that the land temperatures respond more quickly to change in temperature. The ocean, because water has a very high thermal capacity, it responds slower, but it also means it's going to retain that change for much longer periods of time. So what warming we're seeing today is going to continue for centuries to come.
You've got a diagram there, Miles, that shows this prediction that you are working with.
This is a really interesting figure. It's showing predicted changes in temperature at the end of the century. So it's got the predicted changes in temperature by the end of 2100, comparing what it was like at the end of last century. And it's colour-coded, so areas where it's red are high temperatures, high increases in temperature. And what this figure is showing, of course, is that temperatures are going up, but what it's showing is something called polar amplification, and that is when you start modifying the Earth's climate, what you tend to find is that high latitudes, so polar regions, tend to respond more greatly than equatorial regions, particularly in the ocean. So, for example, you can see the North Pole here, temperatures are predicted to increase by 6 or 7 degrees by the end of the century, much more than the predicted increases around the equatorial regions. And even around the Antarctic, you're also seeing an amplification of that temperature change. So we're predicting by the end of the century, the Antarctic waters will be warmer by maybe 4 or 5 degrees. Antarctic waters are super interesting in terms of temperature. It's the coldest temperatures on the planet. So seawater around the Antarctic is around minus 1.9 degrees Celsius.
And it can be that cold because of the salinity, is that right, Miles?
Yeah, so as you know, fresh water freezes at about 0 because it's got no salt in it. But as you add salt, the temperature at which it freezes decreases. And it's about the salinity of the oceans, 34, 35 parts per thousand, means that freezing temperature is about minus 1.9 degrees Celsius. So it's it's that slightly colder. And if you go to places like McMurdo Sound is a large embayment on the Antarctic continent, pretty much due south of New Zealand. It's where the Scott base, the New Zealand research base is. That's the farthest south any ocean goes on the planet. The ocean doesn't go any further south than McMurdo Sound, and it's the coldest water on the planet. In fact, it's so cold that the seawater is always freezing. So you can see this is a picture of a diver here, and you can see some sea urchins here. Here's the diver up here. There's some sea ice. So this is about 10 metres below the surface, and you can see this rock wall here underwater is all covered in ice. It's called anchor ice. And if you look a bit closer, there's the seafloor here, got a starfish, but you can see these large sheets of ice. This is all anchor ice. Seawater is so cold that ice will spontaneously form on any surface, and that includes rocks. It also includes things like sponges. So this is a sponge here. It's about 1 foot high. It's a sea anemone here. But if you look carefully, you can see the sponge is covered in all this ice. In fact, this sponge is probably doomed because that ice is going to freeze. That ice is going to grow. When it freezes, it actually gets rid of salt. So this is actually freshwater ice, and that'll lift that sponge off the sea floor and kill it.
But these animals and plants must have evolved ways to deal with this, to be able to have a whole ecosystem down there. Is that true?
Invertebrates don't have to worry about freezing because they're what we call isoosmotic, so they're the same salinity as seawater, so they won't freeze. But there are other problems. There are other species which are very prone to freezing. That's fish here. So we've got here a fish, and actually you can see some of our experiments, Steve, on the sea floor here. This is about just on the sea floor at McMurdo Sound, and you can see all the ice forming on our experiments. So they affect all our experiments. But yeah, animals do have to deal with that ice, and in particular the fish. The fish have blood which is not as salty as seawater, so they're at a freezing point. They're at a potential to freeze because their freezing point is warmer than the seawater. So they have to have antifreeze proteins. And they're what we call poikilothermal. Poikilothermal means cold loving. They're adapted to cold temperatures. They don't mind the cold temperatures. They've got antifreeze proteins. They don't freeze. But they're also what we call stenothermal. And stenothermal means an animal which is adapted to a very narrow temperature range. And they're stenothermal because not only is the Antarctic the coldest water in the world, but it's also the most thermally stable. Temperatures in the Antarctic don't change at all seasonally. So this little fish here, it's quite happy living in cold water. That's always very stable. It never gets warm. It may be warming by maybe 0.1 of a degree over a season.
And it stayed stable for a very long period of time. They've evolved in this very stable.
Exactly. So these fish have evolved over the last, 4 or 5 million years. They've evolved in these very stable temperatures. They're quite happy at those temperatures. Of course, now temperatures are warming, and one of the issues for that fish is not only it's going to be subjected to warmer temperatures, but those temperatures are going to start varying seasonally. Lots of scientists trying to understand how these fish, for example, will respond to the warming, but also this change in seasonality. They're some of the most extreme and interesting fish on the planet. Here's just an example. This is something called an ice fish. It's transparent, so this isn't an illusion. This is a fish that's transparent, and it's transparent because it doesn't have any hemoglobin. And it doesn't need hemoglobin because it's living in water which is so cold that it has very high oxygen concentrations. Oxygen concentrations that are so high that it can get by without red blood cells. It can just use the blood serum to transport oxygen.
So the colder the water, the higher the oxygen?
Yeah, so oxygen concentrations in polar waters are twice as high as they are in the equatorial waters.
So another problem with global warming, or the warming of the polar ocean, is that there'll be reduced oxygen availability.
Yeah, so oxygen carrying capacity will decrease because it's a function of temperature. And we do know that parts of the ocean are becoming hypoxic, but this will be more of a chronic sort of effect of warming in the polar regions. Yeah, as the temperatures warm, that oxygen concentration will decline. And if you look at the blood of this fish, you can see it's clear. This is the blood of this fish here. It's got no red blood cells in it. This is the blood that you would see in a normal fish, lots of red blood cells like we've got. And that's its gills, transparent compared to a normal fish. So this fish here is going to face all sorts of issues around warming and in particular around how it's going to deal with reductions in oxygen. And you can see it's just not very well adapted to a process which could reduce oxygen. One of the fun parts about going to the Antarctic is that you do get to dive, and we did a lot of diving in the Antarctic. My speciality is actually working on starfish and sea urchins, and as part of that work we would be interested in diving down to collect these urchins, and we were putting experiments out. Like anything, diving in cold temperatures has its own issues. You can see here we're all kitted out. This is me here all kitted out in a complete dry suit. Underneath that dry suit I've got a suit called a weasel suit, which is like a sleeping bag kind of material to keep us warm. Dry gloves. I'll be putting on a full face mask in a minute before I get in the water. And if you look carefully, you can see our dive tanks we've got completely split air supply because the air supply can freeze up in the cold water, so we have to have two independent systems in case one freezes up. This is in the dive hut. The dive hut's heated, it's nice and warm. But in the floor of that dive hut, we've got a false floor. There's the sea ice here, and they've drilled a hole for us so we can jump in the water. and that's what it looks like below that hut. Here's our hole that we've drilled. We're diving through about 2 metres of sea ice, so about from the floor of your house to your roof would be solid sea ice, and then we pop through that, and we've got dive lines and we've got strobe lights so we don't get lost. Diving completely under an ice canopy. The good thing is it's extremely clear water. The other water in the Antarctic is the clearest seawater on the planet, and on a good day you could probably see 500 metres, even up to 1 kilometre underwater if you get the right lighting. So it's unbelievable. It's like diving and floating in air. You just can't see the seawater. We'll get in the water. We'll be in there for about half an hour to 40 minutes before we get too cold and pop back up. More recently, though, we have been using ROVs to collect animals, just because diving does take a bit of effort. So we've adapted our research using ROVs, and this is just another dive hut here. You can again see the more.
So this is an ROV, is a remote operated vehicle. It's got its own autonomous or cable system, and you control it like a robot sort of thing.
Exactly. It's a little underwater robot that's tethered here. So here's the little unit here. It's not much bigger than a lawnmower. It has a couple of cameras. It has thrusters. We've put a little collecting cage on the front of it. Here it's got lights and it can measure temperature and what have you. That's its umbilical. It's a fibre optic cable. And you just fly it like you would a Nintendo or a little home sort of PlayStation. You have a couple of knobs and controls to turn lights on and off and move cameras. But it's very simple to run, very easy.
Right.
We send it down to the sea floor, and we're collecting these starfish here. These starfish is a species called Odentesta validus. This is our aquarium at Scott Base, so this is at the New Zealand Antarctic base. And these are all the starfish that we've collected off the sea floor. And we're just accumulating these, getting these ready to fly back to New Zealand, because the experiments we're running at Scott Base, or running in the New Zealand Antarctic program, are we're looking at long-term acclimation. So we're interested in knowing how well these starfish could acclimate to changes in temperature over the long term. And that means that we can't really do the experiments in Scott Base. We can't stay there for 2 or 3 years at a time. We just couldn't do it. We've got...
So the image on the right is the experimental setup at the Portobello Marine Lab. Is that right, Miles?
Yeah, that's right. So when we collected 600 starfish, and then we flew them up to New Zealand. So this is the big cargo plane coming back from the Antarctic, and these orange boxes here are our starfish flying back. And then they eventually make their way to the marine station at Portobello, and these are controlled temperature cabinets. And if you look carefully, you can just see the starfish inside these little aquarium. Each one of these fridges is temperature controlled. We can say, for example, have this one at +1 degrees above what they were experiencing. This could be warmer by 2 degrees. This could be warmer by 4 degrees, etc. And what we're doing is we're putting the animals in these fridges, and we're warming them up, and then we're looking to see how they respond in terms of how well they grow if they're warmed, how they allocate energy to reproduction, how they allocate energy to growing, how they allocate energy just to maintaining body function. So what we're using, we're interested in the energy budgets. And energy budgets, you can model energy budgets to see, you can start modelling how you would predict an animal to warm and respond to warming. So it's applying real experiments and modelling information to start modelling responses.
Okay, so the temperatures are your treatments, and they are representing those future scenarios that you showed us early on. And I guess you have multiple replicates in each cabinet.
Yeah, that's right. So yeah, it's whenever you do an experiment, you need to think about the treatments. So we went and looked in the literature and looked at what temperatures are predicted for the end of the century. So we want to set up these realistic scenarios. So we'll have your control temperature. So this fridge here could be the temperature of the Antarctic. So that's our control. The animals have come from cold temperatures, and we're going to keep them at those cold temperatures. And then we've decided our next treatment will be +1 degree and +2 degree, because that's what it's going to be like in another 50 years. And then our next treatment could be +4, because that's what we think it's going to be like at the end of the century. And then there's our treatments. And then, of course, because there's variability in any system, and because we want to make robust predictions and make sure our experiments are robust, we need to make sure that we replicate our treatments. So we've got independent replication of our treatments so that if we do see a result, we're sure it's not just due to something funky going on with a particular tank, or there's just so much variability you can't pick up a difference. So if you look carefully in these fridges, you can actually see there's multiple tanks. These are all independent replicates of that treatment. And these animals might survive, but they may no longer grow because they're using all their energy just to deal with that temperature increase. Or they may survive the warming, they may still grow a little bit, but they may no longer have any energy to reproduce. And of course, in the long term, that's not helpful either. So we're just trying to understand, it goes beyond just knowing if they survive or not, but what's the mechanism for their response in terms of they might survive, but they're no longer growing or they're no longer reproducing.
And also, I understand you're also interested in temperature tolerance at different stages of the life cycle as well.
Yeah, so that's right. So these animals, these starfish here, they reproduce via a planktonic larval stage. And one of the important things we need to understand is what part of the life cycle is most sensitive to temperature. So, for example, the adults might be fine. They might like a bit of warming, but what about if their larval stages don't like the warming? You know, what's the critical part of the life cycle? So you're right, we do experiments. Yes. I'll go back to that previous slide, but you can see here, these are just developmental stages of these larvae. These are the sorts of these starfish. These are the sorts of developmental stages that you would see in the plankton that would swim around for a bit, then they metamorphose into an adult. So what we do is we do experiments to try and define what's the temperature range or the thermal window of these different life history stages. So what's the thermal window of the adult? What's the thermal window of the larval stages? And for something like the larval stages, we can use these thermal blocks. So what this is an aluminium block. We heat one end up and we cool the other end, and we set up a thermal gradient, so from warm to cold. And then we can put those embryos or those starfish larvae in these little jars here. And so some will be warming, some will be in cold temperatures, and we can see what's the temperature window, what's the temperature range in which the larvae survive. Yeah, that's just the thermal window, the thermal blocks.
So if I'm looking at this experiment, Miles, the one on the right there, block one, at this end you've got one temperature, and those 4, 1, 2, 3, 4, they're replicates at that temperature, is that right?
Yeah, exactly. So in fact, as you can see, we've got 2 blocks. Yeah, so there's actually 8 replicates. So all this jar here, these are all larvae that are independent replicates subjected to the same cold temperature. And then the second row here, they'll be subjected to the same temperature, etc. So by the time you get up the top, there's our replicate of the warmest temperatures. Yeah, so again, it's a replicated experiment. Yeah, and then with each of those jars, we've also got probably 50 to 60 embryos as well, because they're microscopic. We're talking about things that are 0.5 millimetre in size.
Right. And then the results from these sorts of experiments, Miles, are showing us, yeah.
So like us, like any organism, you have a thermal window or a range of temperatures in which you can survive in. So here we've got cold and warm temperatures, just to say this is some parameter of fitness or health. So there'll be parts where it's too cold for us or an animal, there'll be part of that temperature range was too warm, and then you'll have this, what you call a thermal window, and there'll be an optimal temperature. So for us, it's about, I guess 20 degrees or something like that. So we're interested in knowing what is the thermal window for the organisms that we're looking at in places like the Antarctic. And then we can see, will temperatures exceed that maximum thermal window in the coming centuries here. So these are just examples. This is some examples of the thermal window for an Antarctic species. So cold to warm temperatures. And you can see they're quite happy in cold temperatures, but once you cross about 2 degrees, you can see that their development or their health drops right off. So this animal here, its thermal window for its offspring is about 2 or 3 degrees. So we could make predictions and saying, at the moment it's 0 degrees, but at the end of the century, if it's 4 or 5 degrees warmer, this animal's no longer going to be able to reproduce because its offspring won't survive. Like in many places on the globe, animals could migrate to high latitudes because temperatures will remain cooler. But of course, in the Antarctic, you can't migrate to high latitudes because you're at the end of the world already. so they have nowhere to go. They're going to lose that cold environment that exists on the planet. The temperatures won't be anywhere on the planet as cold as they are now by the end of the century.
Right.
And this is just an example for a New Zealand species, actually showing the low and the upper thermal window. So we would predict temperatures in New Zealand waters when they exceed 20 degrees. For this particular species, this is a Philastus, a sand dollar, that they wouldn't survive in that particular area, and they would have to maybe migrate to lower latitudes where the temperatures are colder. The other thing about the thermal windows we're interested in is, do these thermal windows change when you add another stressor in? Because not only are the oceans warming, but we know that they're acidifying, we know that oxygen concentrations are changing, there could be pollution, and it could be that even though temperature doesn't change, their thermal window could shrink, which would be another interesting thing that you might want to look at. And we've been certainly doing that, looking at whether ocean acidification changes their thermal window and shrinks it. The offspring of these animals, actually, we're finding something called a carryover effect, and it's not great. What it means is that if you stress an adult out and then they go and reproduce, because the adults have been so stressed, they're producing less strong offspring. It's a carryover effect. The adults have been dealing with their own stress, they've had less resources to put into their offspring, and so those offspring are less fit.
So the results you're getting, how can this go to inform, the wider response to this global warming problem that we're facing as a species?
I guess what we're interested in knowing is tipping points. Are there particular critical temperatures? So at the moment, polar species are living at about -1, -2. If you allowed a bit of warming, maybe up to 1 degree, 2 degrees, this particular organism, this particular life history stage, is not going to, suffer too badly. But there seems to be a real tipping point. So what we could say, and as long as, if this feeds into a much broader scientific outcomes, we could feed in and say, look, you've got to try and restrict warming to less than 2 or 3 degrees. That's a real tipping point. So we'll give a target for, for the IPCC, International Panel of Climate Change, to put to the government, look, we need to moderate carbon dioxide concentrations so that we reduce warming to above a certain point.
Yeah, so a bit of a parallel, Miles. Like we know the science behind viruses, and that has informed good governmental and cultural action to reduce COVID-19 problems. I guess the same thing here. We have to listen to the science when we put in regulation or action from a national or a society point of view. With this information in mind, if we are going to have a successful outcome.
Yeah, so that's right. So there's a lot of information, a lot of modelling that looks at if you increase carbon dioxide by a certain concentration, this is the kind of temperature that you would see. The RCPs, representative concentration pathways, and there's business as usual. But those sorts of modelling exercises will tell us, look, if we can restrict our carbon dioxide concentrations to X, we will hopefully restrict our warming to a certain level. So this is how we can guide policy in terms of, look, you really need to target this level of carbon dioxide output reduction, because we know the ecosystems, we've talked about polar species today, but you could do the same for tropical species. They will have very similar tipping points. That if you go beyond a certain temperature, then that's where the issues are going to really tip. So we've got to try and keep our activities so the temperatures don't cross some of these key thresholds.
I'm getting the idea, Miles, too, that temperature is an absolutely critical factor to understand the ocean and ocean life, and because of the ocean's dominance on the planet, to understand survival on the planet.
Yeah, so you're right. I think of all the current stresses in the ocean, temperature is the big one. It's water temperatures around New Zealand and Australia have increased by 2 to 4 degrees in the last 40 or 50 years due to changes in the East Australian current. We know that we've seen big die-offs of of species along the Tasmanian coast. There's been big shifts in species ranges. Species ranges are controlled by temperature. Yeah, temperature is the big issue in the ocean. We're seeing marine heat waves. New Zealand had a marine heat wave a couple of years ago.
Oh, well, gosh, hey, thanks, Miles. That's covered a huge field in a very informative and detailed way. And I can clearly see from what you've said how a focus on one of the polar regions is critical to our understanding of it. Yeah.
No worries.
Thanks very much.
My pleasure. Yep, we'll see you later.